Okay, just to close this chapter, we are going to describe some chambers or cavities that are within the brain, and they're called the ventricles of the brain. Before describing that, sorry about that. Before describing that, I'm going to show it to you so we can be kind of oriented or where they are. See, this is the anterior view. Then we have, see that they look like those horns? Those are cavities or chambers. Inside, they are filled with cerebrospinal fluid. So all of that. And you can see in here, this is like a posterior view, well, like, diagonal. We have one on either cerebral hemisphere. These are two. We have another one in here. This is the third. And this one is the fourth ventricle. And they are connected each other by, can you see these orifices, these ducts? So these are the interventricular foramina that connects the lateral ventricles with the third, and this is the aqueduct of Sylvius or mesencephalic aqueduct or cerebral aqueduct that communicates the third ventricle with the fourth ventricle, and then the fourth ventricle continues with the central canal of the spinal cord. Okay, and all of that is filled with cerebrospinal fluid. So in here, I'm going to show you, which is the image that you have in your book. I'm going to show you this. See that we have one ventricle chambers, that they have a space inside and they're filled with fluid. That's what it is. So we have one on the right and the second in the left cerebral hemisphere. So these are the lateral ventricles. Then we have a third ventricle. And this third ventricle, this is in the lateral view, is located at the same level of the diencephalon. And the diencephalon. And inferior to that, we have the thickest, wider fourth ventricle. And the fourth ventricle is located at this level, okay, in between the pons anteriorly and the cerebellum posteriorly. Now, these chambers are communicated. So in here, there is an orifice, and in here, another one that communicates that. And those orifices are called interventricular foramina, if they're both, and they communicate the lateral ventricle with the third ventricle. The third ventricle communicates with the fourth in the cerebral aqueduct, also called mesencephalic aqueduct or aqueduct of Sylvius. And the fourth ventricle is continuous down here with the central canal of the spinal cord. Now, at the fourth ventricle, we have three openings. We have these two lateral apertures, and we have one median that I don't see. Oh, here it is. Well, I just wrote on that. But a central opening, median aperture. The thing is that the median and the two lateral apertures, they communicate with the subarachnoid space. Okay? So let's see how this fluid flows and who produces this fluid. So first of all, which color should I use, guys? Let's use a green. I don't see any green in there. So first of all, in here, number one, actually, let's start with telling you this is a sagittal section, and you can see here one of the lateral ventricles. Can you see it? Right there, lateral ventricle. This, in between the hypothalamus, this is the third ventricle, and this is your fourth ventricle. That's the median aperture, okay? So both laterals, the third and the fourth ventricle. Now we start right here in the roof of the diencephalon, the roof of the thalamus. Remember that I told you I want you to remember these. Uh, the roof of the thalamus, of the roof of the encephalon. This is the choroid plexus, which is written in here. The choroid plexus is located, in the roof of the first ventricle, the floor of the fourth ventricle, but also in the roof of the diencephalon. And these choroid plexus they're just capillaries. They're just blood vessels, but they're kind of special. They have certain characteristics, and they filtrate the blood, okay? And the blood that is located in here filtrates only the plasma, and that's what it is. This the cerebrospinal fluid. So it's a filtrate of blood. It comes from blood. The thing is that we live in the capillaries, the bigger molecules, proteins, red blood cells, all of those structures stay in the capillary. In here it just comes the liquid part, and which is the plasma, and certain ions like sodium and glucose for sure, because that's the fuel of the nervous system. So in here we produce the cerebrospinal fluid that it fills both lateral ventricles. From there it goes through the interventricular foramen and fills the third ventricle. From there it goes passes through the cerebral aqueduct, reaches the fourth ventricle, and from there part of the fluid gets or flows to the central canal of the spinal cord. The rest of the fluid passes through the median and the lateral aperture to reach the subarachnoid space around the spinal cord and around the entire brain, okay, and bathes the entire central nervous system, and in a way that now the brain and the spinal cord are floating in this liquid that avoids this tissue to being crushed by its own weight, protects from external sources of or the shock's absorption. It nourishes the nervous system because it contains sodium, other ions, and glucose, and gases as well. It also removes waste from the metabolic processes of this tissue, of the neural tissue. So those are the functions of the cerebrospinal fluid. Now, cute and cool fact: the cerebrospinal fluid at any given moment we have around 150 milliliters of cerebrospinal fluid on any given moment. Okay, so if I want to calculate your cerebrospinal fluid, it will be around this number. The thing is that we produce 500 daily, 500 milliliter a day. The choroid plexus produces that amount of fluid a day. So how can you explain that we produce 500 a day, but we only have 150 milliliters a day? And the explanation relies in the fact. Remember these structures that I told you? Hey, pay attention to this. This part of this liquid is reabsorbed into the general circulation. When this liquid, the cerebrospinal fluid, is flowing through the subarachnoid space, remember that the arachnoid peeks into the sinuses, the dural sinuses that act like veins. So this is, in here we have in the blue thing in here, this one that I'm putting in black now, we have blood, deoxygenated blood, venous blood that is going to empty to the heart and to reach the general circulation. Now, when the arachnoid invaginates or evaginates actually towards the dural sinuses, part of the fluid is reabsorbed into the blood contained here in the dural sinus. And from there, it passes to the heart and it gets to the general circulation. And that's how remember that the cerebrospinal fluid is removing waste products from the metabolic processes of the nervous tissue. But it will be done to recirculate all of those waste all around and around and around all the time. So by empty that liquid into the dural sinuses, the cerebrospinal fluid gets rid of those waste products, renewing every single time it passes through here the content of the cerebrospinal fluid. So that's it. Yes, we have concluded the study of the central nervous system. Oh! So finally, now we are ready to discuss the rest. We still have 2 more chapters to go, but they are short chapters. The next one, not that short, but the last one is very short. And I'm going to make it shorter. So peripheral nervous system and autonomic nervous system. See you, enjoy, review, understand this amazing and be amazed of this beauty. See you, love you. Take care, alligators. Too much celebration, and I forgot to tell you this part. I just want to make a quick comparison in here. Look, remember that in order for us to administer epidural anesthesia, you can do it in any segment of the vertebral column, and you just need to pass through the skin, the subcutaneous tissue, and then you pass through the ligaments in between the vertebrae, of course, the posterior posterior is around here, longitudinal ligament, and this is the anterior longitudinal ligament. Okay, now in here, if you want to now perform a spinal tap, to get the purpose of the spinal tap is to introduce the needle into the subarachnoid space, not into the epidural space. So you're gonna go a little bit deeper because you want to get a sample of cerebrospinal fluid to test for tumors, for infections, meningitis, etc. And for that, look at the layers that you're gonna pass by. After the ligament, you go this in here. This is the duramater. The space is the subdural space. Well, I forgot here. This is the epidural space, the duramater, the subdural space, this orangey or pinkish, that's the arachnoid, and then you get right here into the subarachnoid space where the liquid is. And then when you start having liquid right here in your syringe, well, you know that you are there. So for that, you have to pass through the duramater. You have to feel the right? Not like in the epidural space that is before the duramater. In here, a little bit more complications might be possible because you're getting closer, but you don't perform a spinal tap where you have a spinal cord. Remember, we said that the spinal cord ends at the level of L2, well, let's say in general, T11 to 12, and L2 and 3. Spinal taps are always performed at L4 or below to avoid damage of the spinal cord. And what we have after that are just roots of nerves that can be, if you touch them, yeah, it can hurt, but you're not gonna paralyze nobody just by touching it. So those are the layers that you are going to cross, or a needle has to pass by if the intention now is to obtain a sample of cerebrospinal fluid. Now we have concluded with the central nervous system. I won't make any more celebrations because I don't trust myself at this time of the day. See you later, alligators.